Laughter is the best medicine. Laughter is the best medicine. And in this lecture, hopefully, we will be getting a lot of laughs. So, again, this is Best Medicine, and welcome back to the second video in our series for AQA GCSE students doing biology and aspiring to become doctors. This is your teacher Noor, and today we conclude the first chapter, cells. In the previous video, we have talked about cells, and we said that all living organisms are made of cells. And cells can be of different types. We have plant cells, animal cells, and prokaryotic cells. And then cells can also become specialized to carry out different functions. And we looked at some specialized cells. For example, we looked at a neuron, a sperm cell, a root hair cell, xylem cells, and phloem cells, right? Now today, we'll be talking about cell division, how cells can divide, chromosomes, what those are, and exchange of substances. So I am very excited for this video. Okay, so without further ado, let's begin. One kind of cell division is when new cells are produced from pre-existing cells for growth and repair, and something called asexual reproduction. Asexual reproduction is covered in its own topic, and we'll come to that later. But for now, let's just focus on growth and repair. Now, this kind of cell division, which occurs for growth, for repair, and for asexual reproduction, is called mitosis. So how does this work? Let me just quickly show you how mitosis would work. So what happens in mitosis is that you're trying to grow. For example, you remember a zygote, right, from the previous video? A zygote then divides into a clump of cells, and then that clump of cells further divides and keeps on dividing and specializing to give you a baby. That's an example of growth, isn't it? Another example of growth is, and you have all been through this very recently, is puberty, right? Puberty. Puberty growth spurt is also an example of growth. So both of these include a lot of mitosis. So how does mitosis work? Mitosis would work is that you would have a cell, a parent cell, and then it divides into two identical daughter cells. So you have a cell and it has its DNA. Now what would happen is that this cell replicates its DNA. So it replicates its DNA, gets two copies of this, and then it splits into two cells. So for example, over here, you get one cell there and one cell there. So the parent cell has now gone into two daughter cells. Now these daughter cells contain the same exact DNA as we started with from the parent cell. So here's the parent cell with a copy of DNA. Then it replicated its DNA into two different copies, and then each of those copies went into the daughter cells, right? So the daughter cells inherit exactly the same DNA as the parent cell. Not only is this used for growth, which is number one, it is also used for repair. Repair of worn out parts of our body or injured parts of our body. So for example, somebody gets injured. Maybe they get a scratch on their arm and some skin cells erode off. So for example, let's draw some cells here. Here's a couple of cells, and then let's say you get an injury here, and then these two cells are gone. So if these two cells are gone, let's remove them. You gotta get cells here again. So this cell will probably divide, and this cell will divide, and you get two cells here. Okay? And that's how repair would work. So this is mitosis. Mitosis is identical cell division in which the amount of DNA as we go from parent cell to the daughter cell is maintained, right? Next up, we have meiosis. So the other kind of cell division is when new cells are produced from pre-existing cells to make gametes. To make gametes. What is a gamete? Do you know what a gamete is? Can you tell me what a gamete is? This is called meiosis. Okay? So let's have a look at what a gamete is. Alright, so remember that in sexual reproduction, we need a cell that has half the amount of normal DNA, right? So this would have half the amount of normal DNA, for example, a sperm cell. and you would have an egg cell that would also have half the amount of normal DNA, and this would occur in humans. And then you get, fertilization, and then you get a zygote that has full DNA. So, how do we get these special kinds of cells? How do we get cells that have half the amount of normal DNA? The answer to this lies in meiosis. Okay? Meiosis. So meiosis is the cell division that occurs in sexually reproducing organisms in their sexual organs, in which half the normal amount of DNA containing cells are made. These cells that have half the amount of normal DNA are called haploid gametes. Haploid for half, right? Gametes for cells that play a role in sexual reproduction. So the way to remember this is that haploid starts with a ha, and half also starts with a ha. So that's a funny way to remember this. Haploid gamete. Normal cells are diploid. Okay, any normal cell is a diploid cell. For example, a zygote is a diploid cell, or a zygote divides by mitosis into many, many different cells that give a baby, and all of those are diploid cells. However, for cells that are playing a role in fertilization are haploid gametes, and we need meiosis for it. So what is the overview of meiosis? The overview of meiosis is very simple. It begins in the sexually reproducing organisms in their sexual organs. For example, in humans, in males, this would occur in testes, and in females, this would occur in ovaries. In plants, meiosis would occur in the anther from male part, and ovary again for the female part. So the way meiosis works is that you start off with a normal cell, a diploid cell, right? A diploid cell will have a normal amount of DNA. So for example, in humans, our DNA is packed into these packages called chromosomes. So we have 23 pairs of them, or 46 total. So we have 46 chromosomes, 23 pairs of them. In a normal cell, this is a diploid cell. Now, what would happen is that this particular cell will undergo DNA replication, just like in mitosis. So 46 will become 92 chromosomes, 46 pairs now. And then this will split into two. So we get two cells that have 46 chromosomes, 23 pairs. But this is the same as mitosis so far. So what's the difference here? Well, the answer to this is that in meiosis, it's going to divide a second time. Okay, not just one cell division. This is so far the same as mitosis. But not just one cell division, but there's going to be another cell division that is going to half the amount of DNA consisting in these two daughter cells that we have made. So let's just call the first division as meiosis 1, right? And then these are going to divide further. So now the pairs, 23 pairs, become individual chromosomes. Instead of being pairs, they become 23 single chromosomes, 23, 23, and 23, 23. So this is the example in humans. You could have some other organism, for example, if you start off with 12 chromosomes, 6 pairs, and an elephant or something, or some organism that they give you in the exam, then it will replicate its DNA, become 24 and 12, and then it will split into two cells that will have 12 and 6 each, and then eventually you get 6 each. No pairs, singular chromosomes. So that is meiosis. The details of meiosis we'll do in a bit, but let's just go over the neat notes and see what it says. So again, the other kind of cell division is when new cells are produced from pre-existing cells to make gametes. Gametes are haploid cells that play a role in sexual reproduction, in fertilization. So this is called meiosis. Gametes are cells which have half the normal number of chromosomes. For example, in humans, normal is 46, so gametes have 23 chromosomes, and gametes would be sperm cell and the female egg cell. They combine with a gamete cell from the opposite sex in sexual reproduction to make a zygote. And a zygote is a diploid cell, it is not a haploid cell. the first cell, which will divide to make all the cells of the offspring. This will divide by mitosis. And yeah, so that's the introduction to this chapter, and we get our first meme. So let's have a look. Biologists, cells multiply by dividing. Mathematicians. Okay, so because we have just briefly mentioned what a chromosome is, maybe it's better to have a look at the structure of a chromosome and how it looks like, what it is made up of, and how it works. So a chromosome, normally a cell would not just contain chromosomes as it is. You would have a cell that has a DNA that exists as a linear thread-like material called chromatin. So let me just quickly show you how this works. Basically, you have a cell, right? And a cell would have a nucleus, and the nucleus ideally should have DNA, right, inside of it. So DNA should be like kind of like this, a helical double-stranded structure. It looks like this and contains genes. Now this DNA actually exists as a thread-like structure like this, okay? Thread-like structure. This is normally how DNA would be present in any cell in your body. This thread-like structure is called chromatin. So normally DNA inside a cell exists as a thread-like structure called chromatin. That's how the DNA exists. But just before cell division, remember cell division? Just before cell division, you need to split the cells. So because you need to split the cells, you want half the DNA to go one way and half the DNA to go the other way. So for this, what you need to do is you need to condense this chromatin into structures that can be split. Okay, you need to condense this chromatin into structures that can be split. So how do you achieve this? Let me show you how the DNA will condense into chromosomes. So here's DNA right now. Normally DNA is like this, right? If you zoom in on this, and we are saying that this DNA exists as a thread-like structure, kind of like this, and that's called chromatin. Now, just before cell division, before we need to split the DNA, this will this will condense and form these structures called chromosome. How will it condense? This will supercoil, kind of like this. Okay, so this is the supercoiling part of the thread-like structure. The thread-like structure comes very, very close to each other and super condenses to form this chromosome. And this occurs just before cell division. So this little cell here, if it's supposed to divide, here's how it would appear under a microscope. Now you will have these chromosomes here. Okay? Going on in the cell just before cell division. So why do we need to do this? Why do we need to condense the chromatin, the thread-like structure, into chromosomes? The answer to this again is because we need to divide the cell, and we want half the DNA to go one way and half the DNA to go the other way. And there's no easy way to pull this thread-like structure apart. So the best way to be able to do this is to condense it into structures that can be pulled apart, and therefore we get the chromosomes. So for example, we have a cell, and then what would happen is that all the chromosomes would eventually line up in the middle for it to divide, and then these will be pulled to opposite poles, okay? Kind of like this. It's being pulled that way, half the DNA, half the chromosomes are being pulled that way, and so on. So eventually you get these two cells that have the DNA that has been moved to opposite poles, and eventually you get two cells when it completely splits in the middle. So for example, if you have 46 pairs here, then you get 23 pairs go on the right side, 23 on the left side, and you get eventually two cells that have 23 pairs each, right? This is going to go over there, and you get two cells that have 23 pairs each. So that's why we need to make these chromosomes. So chromosomes are condensed chromatin, condensed DNA, which are made just before cell division, so that the DNA can be split into two cells, can be separated into two cells. Let's read this through. DNA of eukaryotic cells exists as thin, linear thread-like structures called chromatin. Just before cell division, however, DNA can get supercoiled or condensed to form chromosomes. If you look into a microscope and you see a cell and you can actually see the chromosomes, that means that the cell is dividing. If you cannot see the chromosomes, usually on the microscope you should not be able to see the chromatin, but if you cannot see the chromosomes in the nucleus, then that means that the cell is not dividing. Okay? Right. So, the majority of cells in your body possess a nucleus, housing genetic material with chromosomes. The chromosomes are condensed strands of DNA molecules. Each chromosome carries numerous genes, so all because the chromosomes have DNA, they have genes, influencing various traits such as eye color. So typically body cells contain 2 copies of each chromosomes, just like we have 23 pairs, and I've already taught you this, and one from the mother and the other from the father. Hence, humans have pairs of chromosomes like pair 1, 2, 3, 4, 5, and so on, all the way to 23. Chromosome pair 23 actually is called the sex chromosomes. They can either be XX or for the female or XY males. So this needs a little bit of a background. Maybe we can use this diagram here. So this lists all the 23 pairs in a human, and this is taken from a human, and this is how chromosomes look like in a technique called banding that we do in the lab. So each of these is a chromosome pair, and here's pair number 1, 2, 3, and so on, all the way to 23. Now pair 1 to 22 are not sex chromosomes, and they do not actually define the sex, sexual genotype of an individual. They therefore are called autosomes. Okay, chromosomes that are normal chromosomes that don't define the sex are called autosomes, while the ones that do are called sex chromosomes. All of these chromosomes again are made up of condensed chromatin, which contains DNA. So each of those chromosomes contain DNA, and on DNA you've got genes. So each of those chromosomes would have genes. Now, sex chromosomes can either be XY, as seen here, which means that that's a male person whose karyotype this is taken from, and if there was another X here, then that would be a female. Okay, for example, over here in this one, actually that's another male. So we have 22 autosomes in this diagram, and then we've got 1 pair of sex chromosomes. This is from a male again, and we also have an abnormality going on here. If you notice at number 5, we've actually got a triplet instead of a pair. Alright, so I hope you understand what a chromosome is. More details on DNA and chromosomes in their own chapter, but this is just an introduction to help you understand cell division. Chromosomes are made up of DNA, which contains genetic information, and we need to be able to split the DNA into 2 cells, which is why we get these, we make these chromosomes to be able to transfer half the DNA one way and half the DNA the other way. And that's really all there is to it. For my main idea for this part here is for you to be able to just understand that chromosomes are packages that contain DNA, and we make them just before cell division, or the cells make them just before cell division, so that we're able to send half the DNA one way and half the DNA the other way. More details on this would be done in its own chapter, which comes later. And yeah, so here's another karyotype. Karyotype is the technique of arranging chromosomes in this pattern, okay? So here's another karyotype. This is actually just a diagram. So can you guess if below karyotype is from a male or a female, okay? And could you please let me know the answer in the comments right next to your nickname, please, so I can follow your progress. And yeah, we get a meme. Doctor, you scored 47 out of 46 on your test. Yay, I finally got a good score on the test. And then the doctor says it's a chromosome test. That moment when you realize your chromosomes outnumber your brain cells. Okay. You know, cells are a lot like humans. Humans, we are born, right? And we grow up, increase in size, increase in our number of cells and our body parts, become bigger, and then eventually we end up reproducing, right? We make more species, offsprings. Similarly, cells, when they are born, they also have this life cycle. Initially, they're just very, very young and small, and then as time passes, they increase in size, they make organelles, they make proteins, and they also replicate DNA. And then eventually, they give off offsprings. They divide. A lot of the cells will divide. So that is covered in a portion called cell cycle, and you need to be aware of the cell cycle for AQA. So the cell cycle consists of 3 stages: stage 1, 2, and 3. Stage 1 of the cell cycle would include, let's get a small cell here. So here's a small cell, and let's assume that this has just been born, right? This is a newborn cell. So this newborn cell, as it enters the first stage, let's call this stage 1, is going to become bigger, and it's going to increase in size. It's going to increase in the number of organelles that it has. It's going to make more proteins, and it is going to replicate, very importantly, its DNA. So DNA will also replicate in this phase. So stage 1 is also referred to as the interphase. And board examiners, they want you to know that the interphase is the longest phase of the cell cycle. And they also want you to know that in stage 1, DNA replication would occur. So DNA replication occurs in stage 1. So now at the end of stage 1, what you get is a bigger cell with 2 copies of DNA. Okay? 2 copies of DNA, because DNA replication has also occurred. Now, once DNA replication is complete, you get a signal for the cell to start dividing, to enter into the next stage. The signal could be a growth factor, a growth hormone. So let's get a signal here. So this could be a growth factor, right? Growth hormone can come in. This could be because of the cell size, that the cell has become big enough that it realizes that it's time for it to divide, right? Or this could be due to injury, for repair, or just growth. Okay, so for any of these reasons, and in certain cases, in cancer, for example, which is a very distinguishable case, it would have no control over its cell cycle. We'll come back to that later. So because of some signal, it enters into the next stage, and that is stage 2. Stage 2 is also referred to as the M phase. M stands for either meiosis or mitosis. So stage 2 is really where the cell division steps would occur. Before that, we have a cell that is preparing itself for cell division. And stage 2 is when the cell division would actually occur. M phase stands for mitosis, which is the cell division of normal cells like skin cells, or it stands for meiosis, which is the cell division to make gametes, to make haploid gametes. And then after this occurs, what would happen is that you get something like this at the end of it. You get cell that is almost split apart with its DNA that has been pulled to opposite poles. So you have 1 copy of DNA on the left and 1 DNA copy on the right, and then this would enter into the next phase. The next phase is called stage 3, and also called the C phase. So stage 3 is also called the C phase, or C for cytokinesis. Okay, this is where the cell will eventually split and give you 2 small daughter cells, 2 small newborn daughter cells, and then both of them will eventually enter the new stage 1. Okay, so again, when a cell is just born, right, a newborn cell, That is the beginning of stage 1. Stage 1 is when a cell has just been born. In stage 1, it will grow in size, it will increase its number of organelles, make more proteins, and very importantly, it will replicate its DNA. So in stage 1, what would occur is that you have chromatin, right? The DNA is present as chromatin. I'm just going to draw it on the right side. So thread-like structures, chromatin. At the end of stage 1, you still have chromatin. And then at the very beginning of the M phase of stage 2, it would become chromosomes. Okay, chromosomes. So now if you look at a microscope, you will be able to see chromosomes inside the nuclei of the cells that are in stage 2. And then eventually it will enter into stage 3 when you're able to see cell that is almost split into 2 halves. And we'll look at some pictures in a bit. Okay, so that is how the stages of the cell cycle pan out. So let's have a read at your text. The cell cycle is the process by which a cell duplicates its DNA and undergoes division and produces 2 daughter cells. It consists of the interphase, which is stage 1, in which cell growth and DNA replication would occur. Very important for you to know is that interphase is the longest phase, and DNA replication occurs in the interphase in the stage 1. Followed by the M phase, which is stage 2, division of the nucleus. And then thirdly, you've got finally cytokinesis, which is division of the cytoplasm of the cell. Okay? Now, for anybody who is not doing AQA and watching this, you might be expected to know some different ways to define the cell cycle. So I wouldn't want you to take this information, okay? Cell cycle can also be divided in different ways other than that mentioned in the AQA syllabus. So this might not be useful for you. Question for you guys: which of these stages is the longest? Quote the question and answer in the comments, please. I want to see your progress, so please also mention your nickname, okay? Alright, so going into the details of this, stage 1, also called the interphase, cells become bigger, they make proteins, organelles, as we've already discussed, and cells replicate their DNA. Now again, this is a cell that is an interphase, and you can see that it's got the DNA in the form of chromatin, right? And not chromosomes. You just see thread-like linear structures, and if you look at them on a microscope, you would not be able to see them. Then you enter into stage 2. In stage 2, the nucleus will be broken so that the chromosomes come out, and the chromosomes also start to appear, and so that you can pull the chromosomes to opposite sides of the cell. So in stage 2, here's what occurs. First of all, chromosomes appear. Remember how chromosomes are formed? Chromatin will condense into chromosomes just before stage 2, or in the very early part of stage 2. So chromosomes appear, and then what would happen is the nuclear envelope, we call it, that surrounds the boundary of the membrane of the nucleus, is broken so that the chromosomes come out. And then those will be pulled apart towards the opposite ends by something called spindle fibers, okay? Spindle fibers. So as you can see in the picture below, you've got these chromosomes lined up and attached by these fibrous proteins, and these proteins are called spindle fibers. Okay? The spindle fibers will shorten, and they will pull the chromosomes towards opposite poles. And eventually you get this particular end result for stage 2, the M phase. After this, you will enter the next phase, the cytokinesis phase, in which the cell membrane and the cytoplasm will divide into 2. So very important for you to take away here is that in stage 2, you're able to see chromosomes. You're able to see chromosomes. In stage 1, you're not able to see chromosomes. Okay, so stage 3. In stage 3 cytokinesis, what would happen is that you will split the cytoplasm, split the cell membrane into half. So in animal cells, the way this would work is that the cytoplasm and the cell membrane split to divide the cell into 2, and nucleus starts reforming as shown here. Basically what happens in animal cells is that you have this invagination of the cell membrane. It's going to go inwards, and it's going to keep on going inwards until it completely splits these into 2 cells. Okay? But in plant cells, it's a little bit different. In plant cells, remember they have a cell wall, right? So you cannot really manipulate the cell membrane all that much because plant cells are a bit rigid. So let's get a plant cell in uh, at the end of stage 2, and you've got the cell wall here, and you got one copy of DNA on the left side, one copy of DNA on the right side. You do not have the infolding of the cell membrane. Instead, what you get is formation of cell wall in the middle of the cell, and then cell membrane in the middle of the cell, and this is called the cell plate. And then this cell plate will complete over time to eventually split the cell into two. Okay, so again, in plant cells, instead of invagination of the cell membrane, the cell wall and cell membrane develop in between the two cells, forming something called a cell plate to split it. Okay. And lastly, for this portion, we have picture identification. First, I'm going to show you some. So let's have a look at S1. This right here, that's stage 1. Why is it stage 1? Because we cannot see the chromosomes, and we can only see one cell. If you look at this cell, we can only see one cell, and we do not see any chromosomes. Then here we have a lot of stage 2 cells. For example, this one here is stage 2. Why? Because it's just one cell, but we can see chromosomes here. Here's another stage 2. Just one cell, but we can see chromosomes. Then this right here is stage 3, because we can see two separate areas containing DNA, containing chromosomes in this stage, and over here as well. This is stage 3. So, question for you guys is, I want you guys to let me know the stages of the yellow marked cell on the left and the blue marked cell. Okay, so that was mitosis. Mitosis, again, is the cell division that is used for growth, for repair, and for asexual reproduction. And it involves the parent cell replicating its DNA and then sending that DNA as it is to the daughter cells, so that the DNA number is preserved and the type of DNA is preserved. It's identical DNA, and therefore the daughter cells are identical to the parent cells. So again, just a bit of a recap. It involves the steps of nuclear envelope being broken down, chromosomes condensing, chromatin condensing into chromosomes, and then eventually moving to the opposite poles by the use of spindle fibers. So some features of details of mitosis. Again, DNA condenses to form chromosomes in the beginning of mitosis. Nuclear membrane breaks down, exposing chromosomes in the cytoplasm. Now, once the chromosomes are exposed, you would have the spindle fibers that come and attach to the middle of the chromosomes and eventually pull them apart to opposite poles. So chromosomes line up in the middle to be pulled to the opposite poles of the cell. Daughter cells will have exactly the same DNA as parent cells. That's very important, and therefore there is no genetic variation. There is no genetic variation. What this means is that there is no differences in the genes of the parents and the offspring daughter cells. That's very, very important for mitosis. Mitosis occurs only in the somatic cells of the body. What's a somatic cell? A somatic cell is a normal body cell. For example, a skin cell, a liver cell, stomach, intestine. The other kinds of cells, which are not somatic cells, are called germ cells. And germ cells are the cells that are special kinds of cells that are present in the testes, in the ovaries, and those undergo meiosis. So remember meiosis from a while ago? Meiosis occurs in germ cells, cells that will eventually produce haploid gametes, and these only exist in sex organs of sexually reproducing organisms. Most of the normal cells of the body are called somatic cells, and they include, for example, skin cells, liver cells, stomach and intestine, and so on. Now, something that I want to mention here is that there are two types of cells that do not actually divide, and these are neurons and muscle cells. Okay, neurons and muscle cells don't divide. So what this means is that once some neurons are lost, you will not be able to get new neurons at that place. And once some muscle cells, specifically, and I want to write this down, actually, this is skeletal muscle cells, the muscle cells that help you move, and cardiac muscle cells. the muscles of the heart. If these cells end up dying in an adult human being, you cannot repair them. And if neurons end up dying, you cannot repair them. Smooth muscles cells are another type of muscle cells, and we'll do more details on this hopefully in the next video. But neurons and muscle cells will not be able to divide. Skin, liver, stomach, intestine, all the other somatic cells are able to divide by mitosis. So you're able to repair those organs if damaged. Okay, so because this section is finished, we get a meme. When your sister steps on your toe. Anybody gets this? When your sister steps on your toe. What's happening here? That's mitosis, right? Mitosis. And next up we have meiosis. So meiosis, we've already looked at the overview. Meiosis is the cell division in which we produce haploid gametes from diploid germ cells. So we'll have germ cell, for example, this is a germ cell. Now before this there would be a germ cell in the stage 1. So you would have a germ cell in stage 1. Let's talk about a human germ cell in stage 1. The human germ cell in stage 1 would have 23 pairs, 46 chromosomes, and then it would undergo in stage 1 interphase DNA replication. And as it enters into stage 2 M phase, meiosis phase, this would have 46 pairs and 92 chromosomes. Then it will end up dividing once, producing 2 cells. So that is meiosis 1, and then those 2 cells will end up dividing to give you 4 cells, right? Those 2 cells each divide and give you 4 cells. That's meiosis 2. So meiosis contains 2 cell divisions. Also, something very important occurs in meiosis, which is that as meiosis is occurring, particularly meiosis 1 is occurring, the DNA, the chromosomes, are actually modified. They are changed a little bit. Some genetic variation is introduced. The mechanisms of how that occurs is beyond the scope of AQA syllabus. However, you just need to know that genetic variation is introduced in meiosis. Genetic variation. Okay, so what I mean by this is that if this is the parent cell, the daughter cells that you get, these 2 daughter cells that you get, these are actually non-identical to the parent cell. Some DNA changes have occurred from the parent cell as we go to the daughter cells. So genetic variation occurs in meiosis. And you have 2 cell divisions in meiosis. And meiosis only occurs in germ cells, not somatic cells. Okay? Alright, so let's have a look. Meiosis involves 2 divisions, meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes, which are pairs, separate, having the chromosome number. So you go from 46 pairs to 23 pairs. In meiosis 2 separates sister chromatids, so you split the pairs now, resulting in 4 unique haploid daughter cells. Meiosis occurs in testes and ovaries in humans. In plants, this would occur in the anther for males and ovaries for females. Meiosis is vital for sexual reproduction as compared to mitosis, which is vital for asexual reproduction. And it ensures diverse offspring because you have genetic variation in this, and that's very important, okay? You must remember that there is genetic variation that is introduced in meiosis, because we will focus a lot on this in the future chapters. So meiosis makes babies, while mitosis makes skin, okay? Germ cells give gametes that help make babies. Mitosis helps repair damaged skin. So here's a couple of differences between meiosis and mitosis. I've listed them down. I've already told you about them, but let's just quickly go over them. You can pause the video, take a screenshot of this, or you can text us in the WhatsApp link provided below to buy these notes, and you can annotate by yourself on the differences and on anything else. So meiosis, this produces sex cells or gametes, which are haploid cells, for sexual reproduction, while mitosis produces somatic cells, normal body cells like skin cells, liver cells, intestinal cells, and so on, for growth and repair. The number of divisions in meiosis is 2. You have meiosis 1 and 2. Mitosis only has 1 division. Number of cells, it produces 4 non-identical haploid cells. So 4 non-identical haploid cells. while mitosis produces two identical diploid cells. Diploid cells, right? Haploid versus diploid. Genetic variation. Meiosis introduces genetic diversity, while mitosis generates genetically identical daughter cells. Of course, for example, if you're trying to repair skin, you want to repair it using similar skin cells. And so you don't want any genetic variation to occur there. The chromosome number halves the chromosome number, haploid cells, while in mitosis you have the maintenance of the same chromosome number. For example, we start from 46 chromosomes, 23 pairs in mitosis, and we get 46 chromosomes, 23 pairs in each of the daughter cells in mitosis. While in meiosis, we start with 46 chromosomes, 23 pairs, and at the very end we get 4 cells, each of which will have 23 chromosomes, no pairs. Okay? Meiosis occurs in the germ cells, in ovaries and testes, while mitosis occurs in somatic cells throughout the body. Role in organisms. Meiosis essential for sexual reproduction and offspring diversity, which is very important. We'll talk more about this in the ecology and evolution part. While mitosis is responsible for growth, responsible for repair, and responsible for asexual reproduction, which we'll talk about in a bit as well. Examples: formation of eggs and sperm, right? That's one example. Egg cells, also called ova, for plural, ovums, singular. While tissue growth, cell replacement, and development for mitosis. Okay, so these are all the differences, and these are very high yield. You need to know them. A 4-marker can come on these, and I hope it comes because you will be ready, right? And again, I keep on emphasizing this, but just knowing the content is not sufficient. You need a lot of exam practice, okay? A lot of exam practice. All right, so we finished that chapter, and we have a meme. This one's from me, I think. The teacher trying to explain the difference between mitosis and meiosis to the class. Am I Patrick right now? I don't know. Okay, so enough about eukaryotic cells. What about prokaryotic cells? How do they divide? Right? Prokaryotic cells, you remember what those are? Bacteria and archaea, right? Bacteria and archaea are prokaryotic cells. So how do prokaryotic cells divide? Well, let's have a look. They divide by a process called binary fission. Binary fission is just a very weird sort of a technical term, but it's just basically you have a cell, right, a bacterial cell, and it divides into 2 identical daughter cells. That's all there is to it. So how will it divide? Well, it will obviously undergo DNA replication first. So let's have a look. Okay, nothing complicated in this. You just need to remember the name binary fission, which is the term that is used for cell division of bacteria and archaea for prokaryotic cells. So you have a cell basically, and you have DNA in this. And before cell division, again, every single time you have to replicate the DNA. So this will replicate DNA. You got 2 copies, and then each of those will go into identical daughter cells. Okay? And that's really all there is to binary fission. So 1 goes into 2, and then 2 can go into 4, and 4 into 8, and so on. And this would occur very quickly, you know. So bacteria are able to divide very, very quickly, which means that if you have a single bacteria, and if it has a good medium, good temperature, good environment, it can very quickly, very, very quickly divide and produce a very big colony. So, you know, you should not leave out food in warm places. You should store your food in colder places where bacteria is not able to divide, because they divide very quickly if they have nutrition. Okay, and let's have a look. So in your need notes, it says that binary fission has these particular steps. Number 1, we have replication. We have DNA replication. So DNA replication would mean that the bacterial DNA is replicated, it's copied into 2 identical segments. And then you'll have cell elongation, becomes a bit bigger. Okay, so DNA replication, cell elongation, then cell division, and eventually you get formation of 2 identical daughter cells. In AQA, they don't really test you all that much on the steps of binary fission. However, they expect you to know that it is identical cell division, DNA replication occurs in this, and it is called binary fission, okay? Especially, it is called binary fission. You understand this? Now, in your required practicals, as well as in the spec, you are expected to know how we can grow our own colonies of bacteria. We can actually grow our own colonies of bacteria by binary fission if we provide the correct conditions. So this is very simple. This is very, very simple, and hopefully we can go through this very quickly. So number one, we have growing bacteria. How can we grow bacteria? Let's read this through, and we'll go through the pictures in a bit. The bacteria can be grown in warm environments. You need warm temperature. Very cold temperature will not support the division of bacteria, which is why we have these freezers, right, which help preserve our food from bacteria, and fridges. Okay, so warm environments with culture mediums. Culture mediums are basically a form of agar jelly. They contain a nutrient-rich solution on which the bacteria can be cultured because they, of course, need nutrients for development. The culture medium is placed inside a petri dish, as shown here in the diagram, and bacteria are added to the culture medium by something called an inoculating loop. You need to remember this term, inoculating loop. Inoculating loop is what's shown over here in this image. Basically, what you do is you dip the tip of the inoculating loop in a sample containing bacteria. For example, and occasionally in hospitals, they do this a lot, where you get a person's urine sample and you want to test if the urine contains any bacteria. So what you do is you take the person's urine, you dip the inoculating loop in the urine, and then once you take out the inoculating loop, you streak it, right, basically rub it against the surface of the nutrient-rich solution, the agar jelly, on the petri dish, as shown in this diagram. So inoculating loop, dip it in the bacteria, then streak it, right, this is called streaking, on the inoculating loop. Next thing you want to do is take that petri dish on which you've streaked the bacteria to allow it to grow, and put it in a warm temperature environment, right? And this is usually done in an incubator, to allow the bacteria to grow. Let me quickly show you. So after you have this inoculating loop, you streak, right? And after streaking, you put it in an incubator. And after a while, maybe 24 hours, 48 hours, depending on the bacteria and how much time you want to give it, you can come back and see small bacterial colonies. And you can test these bacteria for different tests. Maybe if you want to identify what bacteria it is, you can try to test to identify the bacteria. If you want to test the antibiotic resistance to these bacteria, you can also do that, right? So it's once you have the culture, you can do multiple things to them. But for you, all you need to know is how you can grow the bacteria. Again, culture medium, key term, warm environment, agar jelly, nutrient-rich solution, petri dish. All of these are very, very key terms. Inoculating loop, streaking. Okay, I've highlighted these terms so that you can remember them, and hopefully you can annotate these on your own notes that you buy from the WhatsApp link. Okay? All right. Here's a meme for you guys. Bacteria in nature eating dirt and defying the physical limits of life. Bacteria in the lab. Yep, this is a very big challenge that a lot of the scientists face. Okay, the other thing in the required practicals is testing the effects of antibiotics on bacteria. Do you know what an antibiotic is? Antibiotics are medicines that kill bacteria, right? That kill the bacteria. So for example, if I were to put an antibiotic here, let's make this a bit whiter. Let's say this is a very rich sort of bacterial, or maybe make it a different color. Something like this. Okay, so let's say I have this bacterial culture and I put an antibiotic into this, right, a red color antibiotic. Now what would occur is that this would start killing the bacteria around it. Okay, start killing the bacteria around it because it's an antibiotic, and you'll see a clear zone forming around the antibiotic. So keeping that in mind, let's have a read of this. How can we test the effects of antibiotics on bacteria? And that is required. So let's get this image here as well. Place antibiotic-soaked paper discs on a bacteria-covered agar plate. So this is an agar plate, and the yellowish thing over here is the bacteria, leaving the space between them. Antibiotic diffusion creates inhibition zone. The inhibition zone, or this clear zone. Resistant bacteria, however, would not show this inhibition zone, and they continue growing, while non-resistant strains, they die. We can also use a control. Control is something that helps you compare the result of antibiotic versus no antibiotic in this case. So that's what's called a control. A control allows for the best comparison to compare the effect of not using an antibiotic. So a control, for example, we can have a paper disc that is not soaked in antibiotic. For example, we can just soak it in sterile water to isolate antibiotic effects on bacterial growth. And we can let the plate sit for a sufficient amount of time at a good temperature. And obviously, the more effective the antibiotic, the larger the inhibition zone against the bacteria would be. Okay? So, I have a challenge for you guys. Why don't you pause the video and answer this question for me, okay? And I really want you guys to answer this. I would be expecting some answers with some nicknames, please, okay? So challenge for you guys. Pause the video, label 1, 2, 3, 4, 5 in this Petri dish, and code the question as well, and with your answers in the comments. And please write your nickname right next to your answer so that I know I can follow your progress, okay? Alright, so once, because we finished this, let's get a meme. Okay, here's Gru. Who likes the movie Despicable Me? I love it. Okay, so here's Gru. He says you grow some bacteria, you treat it with antibiotics. Okay, the bacteria resists the antibiotics. Wait, what? The bacteria resists the antibiotics. And that's a problem. Also, a very popular question on the board exams is how can we prevent the contamination of the cultures? Growing bacteria must be done in a very sterile environment because we want to isolate one kind of bacteria. If there's multiple kinds of bacteria, that can hinder our results and our tests. So we want to do it in a very sterile environment, make sure that we only have one type of bacteria. So how can we do this? Remember, contamination can hinder results. So how can we prevent contamination? And this is a very popular question. I've seen this multiple times on the AQA board exams. So one of the things that you can do is close off the lid from the top of the Petri dish to prevent microorganisms entry through air. Another thing that you can do is heat the Petri dishes to kill bacteria inside, right? This is also a very popular answer. I would go with this. Pass inoculating loop through a hot flame to sterilize it before you dip it in the solution, for example, urine or blood or anything else. So again, sterilize the inoculating loop. Put the Petri dishes upside down in the incubator to prevent condensation droplets from interfering with the bacterial culture. Okay, I think I would go with this answer first, and then this answer second, and this answer third. Okay, usually you would be asked to give maybe 2 or 1 reasons or methods to prevent contaminations. Okay, now I expect you guys, because you guys are watching these videos and you want to become doctors, I expect you to also be good at math, because math is very important for medicine. Basic math, at least, is very, very important for medicine. So mathematically comparing the inhibition zones, remember the inhibition zones, right? So how can we mathematically compare the inhibition zones, which again is this area here? How can we mathematically compare the inhibition zones? But of course we can do that by finding the area of the clearing zone, right? Area of the inhibition zone can help us compare. So for example, we can measure the diameter of the zone with a ruler and divide the diameter by 2 to get the radius. And then we can do pi r squared to calculate the area. So for example, if you have a Petri dish, and in that Petri dish you have you have a culture of bacteria, and you put two antibiotics into this. You put antibiotic A and antibiotic B, and you find that the diameter of the clear zone that is formed for antibiotic A is 10 millimeters, while for B is 7 millimeters. So you can compare the area using pi r squared, and you get this ratio, 100 pi, ratio 49 pi. Okay, and this means that A is the better antibiotic. Okay, a similar approach can also be used to compare size of bacterial colonies, although not very popular on the exams. Okay, I have another question for you guys. Okay, there's a lot of questions, but I want you to participate. I love it because I'll be reading all the comments. So here's another question for you students, and this is sort of like a personal one. You might have to think a lot about this. What is your favorite bacteria, and why? Okay, take your time on this. You can do your research and quote this question, please, because there's so many questions, and add your nickname. Okay, so for example, my favorite bacteria is pneumocystitis carinae, because it does not affect healthy individuals and only causes disease in severely ill patients with AIDS. Okay. Alright, so that was relatively straightforward. Basically, what you need to do is there's a bunch of information that you need to memorize about culturing bacteria, what is required, and what is the method. Again, everything is in the video, and you can watch this video over and over and over again. Also, it's in your notes, so you can review the notes over and over and over again. So make sure you do that, and moving on to the next part, which is completely different from the previous part. Growth and differentiation in humans. Now, you might be questioning as to, well, we've done differentiation already in the previous video. Yes, we have, but not in that much of a detail. That was just an introduction, right? So we kind of do it again, and we look at some differentiated tissues, okay? So growth and differentiation in humans. Well, remember we have haploid gametes, which were formed in meiosis. They fuse together during sexual reproduction to form a diploid zygote, the first of many cells in an offspring. Now, remember, if this is a sperm, okay, over here we have the diagram. Anyways, here's the sperm and here's the egg, and they fertilize and we get a zygote. Right, and then this zygote divides into a clump of cells, and these are the embryonic stem cells. And these embryonic stem cells can form an entire human being, a baby, right? So because they're able to form a baby, but they can form all the cells in a human being. And so embryonic stem cells are able to form all kinds of cells in the human body, ranging from skin cells, muscle cells, liver cells, even neurons and skeletal muscle cells. Remember, normally we're not able to divide muscle and skeletal muscle cells, cardiac muscle cells, and neurons, but embryonic stem cells can form these, of course, and that's where they come from. So the ability of a cell to become a different type of cell, for example, we're going from an embryonic stem cell to skin cells, muscle cells, neurons, is called differentiation. Again, because we already covered this once, this should be a good revision for you guys. So how does differentiation occur? I want to read this. Recall that because all cells derived from the zygote contain the same DNA, they should ideally have the same genes and hence the same functions. So how do they have the same DNA but different functions? The answer to this is because, again, they are turning different genes on and off. For example, if you have one big, it's kind of like having Windows on a computer. You know, Windows on a computer contains everything. But if you want to play a video game, you just turn on that video game and close everything else. If you want to use Google Chrome, you open Google Chrome and close everything else. Similarly, DNA is kind of like Windows. So DNA would have all these programs, all these genes that perform different functions for different cells. So here's maybe this portion performs liver cell function, right? And this performs function of a neuron. So what you do is, in a liver cell, you turn this on and you turn this off, right? And turn everything else off. So that's how DNA works, kind of like Windows, and you turn on certain programs and turn certain ones off. So the reason why that's not the case, that they don't perform the same functions, is because During differentiation, certain genes are turned on and certain genes are turned off. And this sequential turning on and off of genes allows cells to have different qualities, right? For example, in a neuron, which you remember a neuron from earlier, which has an axon, dendrites, right? Cell body. Genes for those things would be expressed. And genes for other things, for example, the sperm cell genes would not be expressed in a neuron, right? While in a sperm cell, genes for flagella and acrosome would be expressed. And therefore, we have different kinds of cells. Now, in adults, we also have stem cells. Not only does the embryo have stem cells, in adults, we also have stem cells. In adults, we also are able to form some kinds of cells, but not all kinds of cells. So adult stem cells can only divide into a limited number of types of cells. Those are called adult stem cells. And mostly no growth takes place. Cell division, differentiation would occur only to replace damaged, worn out cells. If simple mitosis, for example, skin cells and liver cells would simply divide by mitosis, replace damaged tissue, is not possible, simple mitosis is not possible, then adult stem cells are present for differentiation. And these include those in the bone marrow. Bone marrow contains stem cells that produce blood cells, which help form different kinds of blood cells like red blood cells, white blood cells, platelets. In the skin as well, we have some stem cells which replace dead skin cells. In the intestines, we have some stem cells, right? In the respiratory tract as well, we have some stem cells. But again, cells in the respiratory tract cannot form all types of cells. Maybe they can only form a couple of different types of cells. Same goes for the intestines and the skin stem cells, as well as the bone marrow stem cells. Okay, so some cells cannot be replaced in an adult, and these include skeletal muscle cells. You cannot replace them. If a skeletal muscle gets damaged, maybe in an accident or something, it cannot be replaced. Cardiac muscle cell, people who get heart attacks, their heart doesn't become better. It doesn't regrow itself because, again, you're not able to do mitosis in them, and you don't have stem cells for them. Same for neurons. Okay? So this is how it occurs in humans, as well as similarly it would occur in plants in their stem cells. So how does differentiation occur, or where does really differentiation occur in plants? Let's have a look. Plants also have stem cells, right? Plants would also have stem cells. And plant stem cells are present in meristems. Meristems are mostly the area of a plant that contain growing regions like the buds or the top of the stem. But really, most cells in a plant have the ability to become meristematic. So plant stem cells are in meristems, where growth occurs. That's the growing part. And meristem cells throughout a plant's life can become any type of plant cells. You can literally take one cell. It's very different from adult stem cells. You can just take one meristem cell, put it maybe in a petri dish, right? You get a petri dish, get a culture medium, put a meristematic stem cell here, and over time, this should be able to give you a plant. Now, meristem cells are used to efficiently clone plants. Yes. How will that work? I'll show you in a bit. Cloning helps grow more plants, preventing extinction of rare species. So, for example, if you have a rare plant, and you get cells from it, okay? You get each of the cells to different petri dishes to grow different plants, okay? You get one plant here, one there, one there, one there, and then you get, a lot of plants from one particular rare plant because you've used its meristematic stem cells to be able to culture them. Stem cells from meristems cultivate identical plants with desired traits, like disease resistance, benefiting farmers as well. So we can also use cloning to be able to cultivate identical plants. So let's have a look at cloning in plants. How does this work? Let's read this through. There's a couple of methods that we need to be familiar with, and this is a little bit important, often tested on the exam, so a little bit important for the methods. Cloning in plants. Cloning produces identical offspring. Numerous plant clones can arise from small leaf tissue piece. Under suitable conditions, almost all plant cells undergo unspecialized mitosis, creating undifferentiated stem cells that can differentiate into various tissues, forming a new plant identical to the parent. So basically what I'm saying is that any cell really. If I take a leaf cell, a stem cell, a cell from a stem, right? They have the potential to become an entirely new plant if I give it the right conditions. But usually what we do in plant cloning is that we take these cells from growing regions because they have a greater chance of becoming an undifferentiated stem cell. Animal cloning is challenging because we don't have this ability. Because we have early and permanent differentiation, we don't have this ability of these normal cells like a leaf cell and stem cell going back to the undifferentiated stem cell state. So, how can we clone our own plants? Well, either we can extract some cells, or we can just literally take some tissues and start growing them. For example, and you've probably heard of this before, plant cuttings. Plant cuttings is very, very popular, and you need to be familiar with this. Here's what you do. You take a plant, you take a growing stem, right, and you cut it off, the original plant. So this growing stem now would contain the same DNA as the parent plant. And you plant this growing stem in the soil, right? You plant this here. And then this will start becoming its own plant, which will be identical to the original plant because it has the same DNA. You understand? So that is how we clone. We can take multiple stems from a big tree and plant as many of these as we like, or as many of these as we're able to be able to clone plants. So one way of cloning plants is by using plant cuttings. Another way, which I just showed you over here, is that we can take meristematic stem cells and culture them in petri dishes to obtain small plants. So let's have a look at that one. Culturing meristematic stem cells from meristems. So what we do is we take some stem cells, okay, and you put them on a petri dish, and then these will start growing provided the right conditions, such as good nutrients and maybe light if required, and a good temperature, oxygen, water, and they will become small plants. And then we can plant these small plants in pots to be able to grow them. Okay, like this person is doing. So she took this particular plant, and she's got many of those now. She's planting all of these would be identical to the original plant because they all contain the same DNA as the original plant. Okay? So again, that's cloning. Two ways of cloning. Plant cuttings and culturing meristematic stem cells from meristems. Using stem cells. How can we use stem cells? And nowadays, I've seen a couple of questions recently. You have 3 different kinds of papers. You have combined science papers, you have single science papers, you have triple science papers, and in some of these papers, this topic is now showing up. So stem cells can be used, of course, for cloning of plants, right? But also we can use them in medicine to treat diseases. For example, stem cells derived from the bone marrow of healthy donors are utilized to replace malfunctioning blood cells in patients. For example, cancer can be treated with a bone marrow transplant. Embryonic stem cells, they hold promise in replacing defective cells in individuals. For example, you could have something like somebody has a degenerated joint, right? And you're not able to grow that joint, and you use stem cell therapy to be able to grow that joint. Normally in an adult, if the joint wears off, you would not be able to grow the cushioning in that joint anymore because you're an adult and you don't have stem cells there. But stem cells nowadays are being utilized to be able to grow the joint tissue for older people. Therapeutic cloning. Therapeutic cloning facilitates the creation of embryos with identical genetic makeup to patients, ensuring compatibility and reducing the risk of rejection when using stem cells for treatment. This is an ongoing research project in many different research facilities and different universities. What they're trying to do is they're trying to make the stem cells from a human being or different human beings work in such a way that you're able to clone that human being's organs. Okay, so that you have this supply of organs that will never be rejected. Stem cells can also be applied to damaged tissues and allowed to differentiate to reform the damaged tissue. And I just gave you an example of this. For example, cartilage in joints. Now, because we have all of this, all these uses, especially the recent research which is utilizing stem cells in very questionable ways, we have ethical concerns regarding stem cell use in research. And some of the ethical concerns that could arise is that status of human embryos. Okay, so the next topic in this video is going to be on the exchange of substances. This is very, very, very important topic for not just for GCSEs, but also for A-levels, for hopefully you guys are trying to become doctors, so for medicine, and just overall very high yield, very important. And this is used in multiple other topics that we'll do later, multiple more advanced topics, and so you need a very good understanding of the exchange of substances, okay? Okay, so diffusion is the passive movement of particles from an area of higher concentration to an area of lower concentration. What this means is that you have these particles that are fluid and that are able to move from one place to another, and if you allow them, or if there is a difference in concentrations in the areas that they are present, they're able to move from the area of high concentration to an area of low concentration. Let me just show you how this works. So basically, let's say you are in a room, right? So here, let's make a room here. So this is a room, and then here's you, and you're standing here, and then you have, you know what an air freshener is, right? You know what an air freshener is? You're holding an air freshener, and you spray this, and when you spray this, what's going to happen is that you've got a lot of particles that come out from this air freshener, right? A lot of sweet-smelling air particles. Now, what happens is that these air particles, these are gaseous, and because these are gaseous, they have these very rapid vibrations in them, and they have a lot of kinetic energy. Because of the kinetic energy, they discover that, okay, there's not many particles here on the corner of the room, and not many particles there, and not many particles there. And so what they do is they start spreading throughout the room. And after maybe 5 minutes, right, these are evenly spread out throughout the room in equilibrium, and the entire room smells of the fragrance. So that's how an air freshener works, and that's diffusion. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. When we talk about diffusion in fluids, we can also talk in terms of tonicity. So for example, if you have a test tube, and let's just get a couple of test tubes. That's another test tube, that's another test tube, and then let's fill all of them with water. So this contains water, and that contains water, and this one contains water as well, right? Let's make this one very rich with solute. So we will say, okay, this one very rich with solute, and this one has a little bit of solute, and this one does not have any solute. So we will say this is high concentration, highly concentrated, high concentration in this test tube. Another way to say high concentration for fluids is hypertonic fluid, okay? Hyper means high, and tonicity means concentration. So this particular test tube's fluid is hypertonic. This is low concentration, right? And so we also say this is dilute. Dilute as compared to concentrated. And this is hypotonic, okay? Hypotonic. And the other one is just purely water. Now, if I were to make the last one similar to this one here, then they become similar in tonicity, right? They're the same in tonicity. If they have the same tonicity, if they have the same concentration, then we say that both of them with each other are isotonic. Iso means same, okay? Isotonic. You might know the word isotope from chemistry. Similar, those are atoms of the same element with a different neutron number. Similarly here we have isotonic. Iso means same, tonicity means concentration, same concentration. Okay? So if mixed, or if present in the same container, particles will go from high concentration to low concentration by diffusion. Okay? While in isotonic you would not have a net movement of particles. So let's read your neat notes. Key concepts, it occurs in gases and liquids. That's where diffusion occurs, because only gases and liquid particles are able to move. They are fluid. Solids are not able to move. Their particles are fixed at their positions, so diffusion does not occur in solids. For example, in atmospheric air, you would have diffusion. Water, you'll have diffusion, right? And we have these test tubes made. As well as across a membrane in living organisms, you could have diffusion. And small molecules diffuse more easily than large molecules. That's self-explanatory. And so on. So what are some of the factors that can affect the rate of diffusion? This is very simple and hopefully, and also very important. So hopefully we can get through this very quickly. Number one, we have temperature. Remember, diffusion is the movement of particles because they are moving, right? Because they have kinetic energy to go from one place to another. This is why it doesn't occur in solids, because those are just fixed at one particular place. So greater the kinetic energy, greater is going to be the diffusion rate. So higher temperatures generally increase the kinetic energy of particles, accelerating diffusion. Number two, concentration gradient. Of course, the greater the difference between the concentrations, the steeper the gradient between high and low concentrations, the greater the diffusion would be. Remember, there is no diffusion between isotonic, for example, solutions, right? Surface area to volume ratio. This is quite important, and we need to try to explain this. Yeah, I've explained this down in a down layer as well, but let me just quickly show you how this works as well. So surface area to volume ratio. Let's say you have a cell, right? Here's a spherical cell. And let's say the cell's volume is 100 micrometers cubed. Similarly, you have another cell. And that's a bit flatter, okay? And let's say this cell's volume is also 100 micrometers cubed. So the difference between the spherical cell and the flatter cell is the surface area. If particles were to enter or exit either of the cells, let's talk about entering, then this one has lesser surface area available. The one on the left, the spherical cell, has lesser surface area available for particles to enter. While the cell on the right, well, this has a lot of surface area, right? Particles can enter and exit through a lot of different places. So greater the surface area, the greater is for the same amount of volume, the greater is the rate of diffusion. Remember root hair cells? Root hair cells have very flat and long extensions called root hair, which increase the surface area to volume ratio for faster diffusion of materials, faster exchange of materials. Okay? So that is surface area to volume ratio. The larger the surface area to volume ratio, it allows more particles to diffuse at the same time. Distance. Diffusion distance is also very important. If you have a smaller diffusion distance, of course, this will mean that the diffusion rate is faster. Size of particles, again, smaller particles diffuse more rapidly than larger ones. And medium. What is it diffusing through? If it's diffusing through air, water, things like that, right? Ketchup, gelatin. Medium, substance through which diffusion occurs. Diffusion occurs more quickly in less dense mediums. Okay? All right. So importance in biology. Why is diffusion so important? Diffusion actually is very, very important in biology. We have a couple of examples that we'll do in a bit. So it is essential for processes such as gas exchange, which we need for respiration. Remember, oxygen combines with glucose to form carbon dioxide and water and energy. We also need to absorb nutrients, glucose, for example. in the digestive system and filtration of blood in the kidneys. So for this, we need diffusion and other forms of exchange of substances. Right, so coming back to the surface area to volume ratio, we have this very important question, which is, why are cells so small? Why are cells so small? The answer might be hidden in the surface area to volume ratio part. So what is surface area to volume ratio? We just looked at this. Let's just have a look at it again, and then we'll go back. So again, surface area to volume ratio means that if you have the same volume, but if you have a greater surface area, right? For example, on the cell on the right has a greater surface area for the same amount of volume, it has a greater absorption, greater diffusion, while greater exchange, right? And a lesser surface area has a lesser exchange of materials for the same amount of volume. So that's very, very important, so keeping that in mind, let's have a read of this. Remember that a cell exchanges materials, nutrients and waste products through its cell membrane. For example, the two cells that we looked at just now. So if a cell becomes too large, the demand on its resources increases, its ability to exchange nutrients and waste becomes compromised. The limitations arise because the cell surface area may not be sufficient to meet the metabolic needs of its larger volume. So cells tend to be smaller to be able to exchange more materials for a given amount of volume. What does this mean? In a nutshell, what this means is that the larger the cell, okay, the larger the cell, smaller or lower is the surface area to volume ratio. So they have a lower surface area to volume ratio, okay? If you have a smaller cell of the same shape, like a spherical cell over here and a spherical cell here, they have a greater surface area to volume ratio. Yep, it sounds fascinating, but that's really how it is. So for example, you could have two spherical cells. Let's just get two spherical cells and try to prove this. So here's a cell that's big, and then that's not a spherical cell. Here, that's a spherical cell. Okay, and then here's another cell. Okay, and then these would have their own radius. Okay, so the radius of this is maybe 50 micrometers. I'm not sure, I'm just guessing, okay? And then maybe let's make this 5 micrometers. So let's calculate the surface area and volumes of each of them. Do you remember the surface area formula of a sphere? Do you also remember the volume formula of a sphere? The surface area formula for a sphere is 4 pi r squared. Okay? And the volume formula is 4 over 3 pi r cubed. So let's have a look at the cell on the left. The surface area for the cell on the left is going to be 4 pi times 50 squared. Ratio 4 over 3 pi times 50 cubed. And this becomes 10,000 pi ratio 16666 pi, which is the same as 1 ratio 16.6. Okay, let's have a look at the smaller cell. So smaller cell has a diameter of the radius of 5 micrometers. Let's have a look at that one. So 4 pi times 5 squared ratio 4 over 3 pi 5 cubed, and that's going to be 25 times 4 is 100 pi ratio 125 times 4 divided by 3 is, which is 100 pi ratio 166 pi. So again, the ratio is much greater essentially for this one as compared to this one. Let's have a look. The surface area over volume for the one on the left is 1 divided by 16.6. As compared to this one on the right, is 1 divided by 1.66, right? Which is 10 times greater than the one on the left. So a smaller cell has or let me just divide this by 10 instead and have a more comparable result. So this would be 10 pi ratio 16.6 pi. So for this one, this is 1 pi ratio 16.6 pi. So basically, you have for 16.6 pi volume for each in the smaller cell. In the smaller cell, you have 10 times the surface area present for exchange as compared to the larger cell. So greater surface area for the same amount of volume is what gives it the edge, a smaller cell the edge in exchange of substances. This is why cells are small, okay? And this is a very popular question. I've seen this on the exam where they ask you not directly but maybe indirectly as to why do we need a transport system, right? Why cannot we just rely on diffusion? And the answer to this would be that, for example, in humans, we need a transport system because we have eukaryotic cells. And eukaryotic cells are larger than bacteria, and diffusion is not sufficient enough, right? Our surface area to volume ratio is low. Our metabolic demands are too high. Therefore, we need to have a transport system to efficiently deliver the nutrients to each of the cells, okay? And I have added this somewhere in your notes as well. So as we finish this, again, I emphasize you will need exam practice and you will need guided practice as to how you get familiar with the exam terminologies, exam vocabularies, the mark schemes that they want you to use, and you'll need a very experienced person who's gone through this and who has gotten the grades to be able to guide you through how to write your answers. Doing the content is, of course, important, but it's not everything, okay? All right, we get a meme. Here's diffusion. Diffusion of molecules from a place of higher concentration to a place of lower concentration until the concentration on both sides is equal. And here's a cat trying to diffuse. Concentration equal on both sides, I think. Okay, here, a dilute solution exists, particles in a higher concentrated solution. All right, I'm gonna head out. This is me right now. This is me learning medicine. Okay, possible question again, why do multicellular organisms need a transport system, and for 2 marks? And the answer to this is because eukaryotic cells in multicellular organisms are large. Because they are large, they have low surface area to volume ratio, which means that diffusion is not sufficient to meet the metabolic demands. And therefore, a transport system is needed, it should be needed, to bring nutrients in and take waste products out. Okay. So the next topic is osmosis.